Good morning, once again, colleagues and all the participants who have joined us in our workshop for today. We welcome you to the Research Capacity Development Unit, University of Johannesburg workshop on formulating hypotheses and designing experiments for natural sciences. Before I proceed, I would like, by a show of virtual hand, for the participants and also for Dr. Mieta to confirm that you can all hear me well and you can see the video that I've shared with you with the facilitator bio, on your screens. Just a confirmation that you can hear me well and you can see what I've shared with you from the side. All right, thank you for the confirmation. I would like to first remind you that this session is being recorded. My name is Dr. Gabriel Monyera. I'm from the postgraduate school, the Research Capacity Development Unit, and I'll be your moderator for today's session. So why are we having a conversation on this topic, on formulating hypotheses and designing experiments for the natural science? So I think the topic for today is an important topic that will equip the participants with the essential skills to develop clear, testable hypotheses and to design rigorous experiments in the natural sciences. It will also enhance your ability to construct well-founded research questions, select appropriate methodologies, and ensure the validity and reliability of your findings. And by attending, I think at the end, the outcome is that the participants should be in a position to strengthen their research design capabilities, leading to more impactful and reproducible scientific discoveries. Let me thank you once again for taking time to be with us in this session. But also, most importantly, I would like to thank Dr. Mieta for her efforts in preparing and facilitating the presentation for today's workshop. But before I introduce our facilitator to you, I would like to go over some housekeeping rules and important information. And for housekeeping rules, the first thing is that if you have any question during the presentation, please feel free to raise your virtual hand. I would now enable your mic for you to be able to speak, or alternatively, you can simply text your question in the chat box. Then I will bring it to the attention of the facilitator to address the question. So that is also dependent on the preference of our facilitator. Whether, I think at the beginning of the presentation, she will let us know whether she prefers questions to be asked during the presentation or during the question and discussion segment, later after she has finished presenting. Then also, like I've said, the session is being recorded and the slides will be requested from the presenter. Both the recording and the slides will be uploaded on our Moodle communities by the end of the week. And during the session, I will send a feedback survey that I would like to please request all the participants to fill it out for us to gain insights on whether this workshop was useful and also how to prepare for our future workshops as well. It will enable us to also draw some kind words that we can provide to our facilitator for hard work and effort. By filling the feedback survey, it will also guarantee that you will receive a certificate of attendance. So let me introduce our facilitator for this session, Dr. Sumaya Mieta. I'll read your bio in your hearing. Dr. Mieta is a highly skilled professional in the fields of medical microbiology, molecular biology, and water and health research. She completed the undergraduate and postgraduate studies in biomedical technology at the University of Johannesburg with distinction in the HPCSA board exam as a specialist microbiology medical technologist. Her expertise spans medical microbiology, research methodology, medical ethics, and technology innovation. Dr. Mieta has extensive teaching and research experience, including facilitating microbiology and research methodology courses, supervising honors and postgraduate students, and contributing to the development of young talent in medical laboratory sciences. She has worked in academic, medical, and research environments, securing funding from the National Research Foundation, Technology Innovation Agency, and Water Research Commission, and has published peer-reviewed articles and presented research internationally. With this, I'm delighted to hand over to Dr. Mieta, who will be leading us throughout this session. We appreciate your time, Dr. Mieta, and expertise, and also for agreeing to facilitate the workshop for today. And we look forward to your valuable insights that you are going to share with us. I'm delighted now to hand over to you, Dr. Mieta. I'll stop sharing my, from this side, I'll stop sharing, then you can share your slides and lead us from here henceforth. Thank you.
Thank you very much, Dr. Moniera, for that welcome and the house rules. I will begin sharing my slides. Please let me know if you can see them.
Yes, Doc, we can see them, but they are not yet on the presenter mode. Yes, now we can see them on presenter mode. I think let's just get one or two participants also to confirm by show of virtual thumb. Oh, okay, I think we can all see. Thank you. You can proceed, Dr. Net.
Okay, thank you very much for affording me the opportunity to conduct this workshop, and I hope that I will be able to achieve all of the outcomes that you had laid out at the beginning of this session. I would like to request that we keep the questions for the Q&A session, purely for the reason that it might be something that you would be asking, and I will be addressing it in the later slides. So if that's okay with everyone, we could use the Q&A session for the robust discussions. So just a bit of background on who am I. Having listened to the academic bio, I'm not all academic. So I come from a background where I am a first-generation academic. I have a passion for medical microbiology, having worked in the diagnostic laboratories, assisting with diagnosing various infectious diseases from human samples. Along with that, I'm also a mom to two very energetic boys. And at the same time, I wear the cap of an academic, focusing on research, as well as a lecturer teaching in the Department of Biomedical Sciences. So that's a bit about me. And I look forward to today's session for us to be able to engage and discuss. I am in no way an expert on the topic, but I do have sufficient knowledge to be able to guide you and give you the basics into how we go about formulating hypotheses and designing experiments. So before we begin, I just want us to give some thought and to see who we are engaging with. Which department are you from, or which specialization or profession are you in that is attending the session today, just so that we can get a feel of who is in the room. I unfortunately cannot see the chat while I'm sharing.
I will read, I will attend to the chat for you.
Thank you.
Yeah, also the participants, they can type in the chat box. then I will read it in your ear.
Okay, thank you so much for that. So while you are saying which department you are from, I also want you to take a few seconds and think about this. If you weren't in this field or this profession, what career would you be in? And the reason that I want us to think about this is that we are always so focused and so driven on meeting targets, meeting deadlines, moving from one step to the next, that we don't actually sit back and think, is this for me? Am I doing what I want? Do I still have the passion to continue in this? Or do I feel like I should be in a different space? And this changes for each individual. Looking at myself, having been at the university as a student, an undergrad student progressing into postgraduate studies, going out to the laboratory and working, at each point, our goals change. And I know that most of you are now here conducting postgraduate studies, but you might be doing postgraduate studies for different reasons. It might be academic progression, it might be for your family, it might be for yourself. We all have different reasons for what we do. And this was a very profound quote that I came across John Dewey, and he said that we don't learn from experiences. And that is contrary to what we always have been told. You learn from your experience. But what we actually learn from is when we reflect on that specific experience. And that is what you also will need to be able to do at the end of your postgraduate studies. It's not what you experience during your postgraduate studies, what you experience while formulating your experiments, conducting your research, writing up your dissertation or thesis. It is reflecting on those experiences once you have completed that actually gives you that insight and knowledge and experience on the entire process. Okay, is there any comments from in the chat?
Not yet, Doc, but I've just indicated that the participants, they can introduce themselves there on the chat.
Okay, so we have a very quiet group.
Let me just say that, let me take the time as well to just confirm if they can hear us. Participants, are we together, the attendees? Can you hear me? And can you see the slides that have been shared with you? Okay, thank you.
Okay, so I will continue. So when we talk about formulating hypotheses and designing experiments, it doesn't start there. It starts with an observation. And an observation is on something that has intrigued you or something that you want to focus on. And as we go about our daily activities, there are so many observations that we make. And there might be something that you have come across and you have asked yourself a question. And that is the basis of a scientific method. Okay. And so we make this observation and you follow that up with a question. And the question is verbalizing your interest using the term or the questions what, who, where, when, and how. Okay. And once we have that, we then can say, okay, let's formulate a hypothesis, which I've highlighted because those are the two factors or two components that we are going to focus on in today's session. A hypothesis is divided into two sections. Okay? And for us to be able to come up with a hypothesis, you need to have background information. So you make an observation and you start asking yourself all of these questions. What is the reason for this? Why is this happening? Where has it happened? Where has it been studied? Or how would people have studied this and looked at it? And for us to be able to give answers to those questions, you need to go and do background research, what we all know as the literature review, okay? And before we get to the literature review as well, your background research could be very broad. It could just be looking it up on the internet. It could be reading an article. It could be looking at previous studies in a scientific journal or a presentation at a conference. Okay, so that all comprises background research because it gives you additional information. And using that is where we come to the crux of the hypothesis, and that is formulating a possible explanation or prediction for this observation that you have made. Okay? And the experiment is the answer or the process to get to the answer of this question. And it is an investigation that you undergo to be able to test this hypothesis. Yes, it agrees. We see the evidence that the hypothesis is correct, or no, it goes against our hypothesis or our expectation of the outcome of the study. Okay. And this is then followed by the data analysis, where we actually interpret the results obtained from the experiments and a conclusion where you would then respond to the hypothesis and say, no, due to these reasons, the hypothesis was incorrect or the hypothesis was correct. Yes, we've obtained this. Okay, so that's just a brief description of what a scientific method is and how all of the different components come together to create a scientific method. So what is a hypothesis? We've heard this term numerous times through the undergraduate studies, in your fourth year, your honors, progressing into the master's and doctoral studies. So the very basic definition given by the Oxford Dictionary is that it is a supposition or a proposed explanation that is made on the basis of limited evidence. So you don't have this vast amount of information available to you. It's limited evidence. And this is the starting point for further investigation, because now you want to build on this to create more evidence and knowledge, okay? Cambridge states that it is an idea, an explanation for something that is based on known facts, but has not yet been proven. So it's based on something that we know. Yes, this exists, but where is the proof for that fact? And that is what a hypothesis is, according to Cambridge. Then we come to the more scientific and the experts in the research field, where Creswell, who in 1994 defined it as a formal statement that presents an expected relationship between an independent and a dependent variable. So it's a very scientific explanation of what a hypothesis is. And we're going to go through the terms independent variables. What are they? How do they work? Okay. And then Leedy and Omrod in 2001 said it is a logical supposition and a reasonable guess. Okay. It provides a tentative explanation for the phenomena that is under investigation. So it's a very well thought out statement. It's not something that you just pull out from a hat and say, oh, this is a hypothesis. It's a guess, but it is a calculated guess based on some sort of evidence, okay? So why do you need a hypothesis? Why can we not just go ahead and say, oh, I want to investigate this, and let me just go and do the experiments and see what I get? The hypothesis is there because it basically guides the research. Okay? It is formulated from the question that you are asking. So you've made an observation and now you have this question. The hypothesis will be that map that guides the research and keeps you on track. Because as you progress with the research, there are various deviations that can come in, and that can take you either in the direction of your initial observation or take you in a completely different direction. Okay, so that's why the hypothesis is so important. It also outlines the objectives of the experiment. And what are the variables that you're going to be using and the expected outcome of the experiment? Okay, so the hypothesis has certain characteristics. And one of them is that it must be either falsifiable or testable. So you can say, yes, we can test it by doing this investigation. Or you can say, no, this is incorrect because of the evidence that we have found. Okay. And it also provides an answer to the initial question. So some of you might be saying, but then is this not the same thing as a theory? There is a slight difference in that a hypothesis is a tentative explanation that can be tested. Okay, and how do we test it? We do further investigations on this. Whereas a theory is an explanation of observations. So it is what you do at the end of a study, where you will observe all of different individuals' interactions, their behaviors in the certain environment, and you then formulate a theory and say, based on this, when this is present in a specific environment, these individuals react in this way. But we do not say, a hypothesis would say, what the individuals will react in a specific manner in the absence or presence of this factor. Okay, so that is the difference between the two. So what defines a good hypothesis? Because we can all come up with hypotheses, right? A random hypothesis. But what makes it a good and viable hypothesis for you to be able to conduct experiments and provide that further investigations and evidence? It is, according to Edgar and Nance, they state that a hypothesis is a falsifiable and testable prediction of how a system will behave under clearly defined conditions. Or an alternative would be that how would a dependent variable react when an independent variable is controlled? Okay. And a good hypothesis allows us to convert an inductive question to a deductive statement. And what do I mean by this? An inductive question is the initial question that you started with from the observation, okay? Where you start with a specific observation or data, and it aims to discover broader patterns or themes or theories, and it generally moves from particular to general. So you saw a specific thing, and now you want to generalize and say, okay, can this be applied, or is this applicable to a larger population? Okay. So for example, an inductive question would be, what are the experiences of individuals living with pain? Okay. So we're looking at individuals who have chronic pain, and we now want to say, okay, so if these few individuals experience this when they have chronic pain, can we generalize it and say that all individuals living with chronic pain will experience A, B, and C? Okay. Whereas a deductive statement is the opposite of an inductive question, in that it starts with a general theory or hypothesis, and then it tests it through specific observations and data collection. So it moves in the opposite direction, from something very general to being more specific. And an example of this would be, say you want to look at the effectiveness of a new drug on a specific condition. So you would design a clinical trial to be able to see and test if that drug improves the patient's pain outcome. Does it reduce that person's pain? Or does it have any effect on their pain? Or is it just a placebo effect? It has nothing, okay? So that is the difference between these two questions, okay? So the characteristics of a good hypothesis is that it should be observable and testable. So that is something that you can test through experiments and collect data. It must be clearly defined. So you must be able to see what the variables are and what their relationship would be to each other, okay? It should also test a specific concept. So you do not want a hypothesis that has 4 different ideas that you want to test. The hypothesis should be the overarching statement that by doing this, you are going to achieve that, okay? It also should be predictive in that it should be able to define an experiment to test an idea. So if I tell you a glass of water, sorry, a glass of water is not, sorry guys, just needed to clear my throat there. So I tell you a glass of water is contaminated. You are able to test that hypothesis. Okay. Is this water contaminated? Does it have different bacteria in it? Are there viruses in it? And you can already see the different types of experiments that you would conduct to be able to prove that hypothesis correct or incorrect. Okay. It should be falsifiable in that it should be possible to prove it wrong. Okay, there must be that possibility of proving it wrong, and you're going to see why we're saying this. So if I give you these 2 examples, and based on the characteristics that we have spoken about, would you say that from these 2 examples, which one is a good hypothesis and which one is a bad hypothesis? I'll give you 2 minutes to read through it, and you can either raise your hand or you can type it in the chat.
I will allow the mics of all the participants for us to be able to answer this question. Alternatively, you can type in the chat box, then I'll bring it to the attention of the facilitator. Thank you.
Let me put the other slide back so you can look at what the characteristics are of a good hypothesis and then link it to the examples. Are you able to identify these characteristics in each of the hypotheses provided? Anyone?
If you'd like to speak, you can unmute your mic and speak.
I'm hopeful that I haven't lost our participants.
I think we still have them in the session. I don't know why the participants today, they are too quiet. Participants, all the attendees, are we still together? Just can you hear us and can you see the slides shared with you? Participants, are we still together? Just show by, okay.
Okay, so they're still here.
Yes, they are still here.
Okay, so we'll continue. I probably have a shy group of participants. It's fine if your answer is incorrect. We're all learning in this process. So please do take part in this discussion, okay? So if we look at these two examples, if plants are given fertilizer, then they will grow taller than plants that are not given fertilizer. Can we test this hypothesis? Is it testable?
Yes,
Yes?
it is testable.
It is testable, okay?
Yeah.
Is it specific? Yes,
Yes,
it is specific.
it is, yeah.
Okay. So it's testable in that you can grow two sets of plants and you can add fertilizer to one and you don't add fertilizer to the other, and you can see what the difference will be. It's specific in that we're focusing on a certain characteristic or feature. We are testing the height of the plant to check that they are growing, okay? Because the hypothesis says they will grow taller. So we're looking at the height, okay? And we're also looking at the fertilizer. If we use fertilizer, it will grow taller. If we don't use fertilizer, it won't grow taller. But it's also falsifiable, because what happens if a plant that does not have fertilizer grows taller than those that have fertilizer? So the hypothesis can be proven wrong in this instance, okay? So it's also, that's what we mean by the term, it can be falsifiable, okay? The second one, aliens control the weather. How are we going to test this? What methods are we going to use to show that aliens actually control the weather? I think it was a very straightforward one that reading the sentence already says it's a bad hypothesis, okay?
I was
It's
also
not.
wondering how that can be tested, yeah.
Exactly. It's not falsifiable, because if the weather pattern changes, how are we going to prove that it was because of aliens and not a specific weather phenomena or a specific pattern? Okay, it's too vague. There's no specificity in this statement. We're not looking at a specific factor. Okay, if it said that aliens control the weather by changing the magnetic frequencies surrounding Earth, that might be a better hypothesis, okay? And there's no basis in existing knowledge. It doesn't build on the scientific knowledge of weather systems, okay? So I hope these examples can help you understand what a good hypothesis is, okay? Are there any questions on this? If anyone feels that they have a burning issue and we need to address it.
I've allowed the mics of all the participants, so if you have any question or anything that you want to contribute to the conversation so far, you can raise your hand and unmute yourself, and then you ask the question. Alternatively, you can type in the chat box, then I will bring it to the attention of the facilitator. Thank you. Let me just ask one question, Doc. I've always heard when students, sometimes during presentations that I also make questions from students on the difference between a hypothesis and expected results.
Okay, so
they ask what is the difference between the two, what you're expecting to get and
okay,
a hypothesis. Yeah.
Okay, so the hypothesis is basically a statement that you are saying that if we do this, we expect this to happen. But it does not say that if we provide these conditions, that this will happen. Because it could be that we don't get those results. Okay, so that is the difference between a hypothesis and expected outcomes, expected results is what we understand from the previous knowledge. Okay, so they are somewhat linked together, but there's a slight differentiation in that we must remember that expected results are not what we will find. It's the possibility of getting those results. And the hypothesis puts it out in a nice manner in that, yes, possibly we will get this, but there's also that chance that we're not going to. And I think my next slide is going to help us understand that in terms of what are the different types of hypotheses that we get, okay? And we have a null hypothesis, which states that there is no relationship between the two variables being studied. So we say that A does not influence B in any way. They are independent. They work totally separately. So if we change A, B is still going to remain the same. Okay. It states that the results are due to chance and are not significant in terms of supporting the idea that is being investigated. In that we say, oh, it was just by sheer luck that you got this result. There's no significance to this. If someone else conducts this study, they're going to get totally different results. Okay, so that's what the null hypothesis states, that it was purely by chance. Okay, it's normally the null hypothesis states the opposite of what we want to predict or what we expect. Okay, so the null hypothesis will say, if students study for 4 hours a week, they have a 50% better chance of passing the assessment. Okay. The null hypothesis will say that even if the student studies for 4 hours a week, they will still not pass the assessment. Okay, but we predicting that with that effort that the students will put in for that specific amount of time, they will be able to pass 50% of that assessment. Okay, so the null hypothesis states the opposite of what we expect or predict. Okay. The final conclusion of the investigator will be to either retain the null hypothesis and says, yes, the null hypothesis was actually correct. Even if students study for 4 hours a week, they still won't get 50%. Okay, they didn't get 50%. And that either supporting the hypothesis or rejecting it can only be done at the end of your experiments. Okay, but there are times where not rejecting the null hypothesis does not really mean that that hypothesis is true. It could be that there wasn't sufficient evidence against that hypothesis to say that it's true or it's not true. Okay. It normally states the assumption to be tested. Okay. It begins with the assumption that the null hypothesis is true. Okay? And it always contains the equals to sign. Okay, so there's always equality in the null hypothesis. Then we have something called the alternate hypothesis. And the alternate hypothesis states that there is a relationship between the two variables that are being studied. One variable has an influence on the other. Okay, so if I'm trying to think of just a straightforward example. If the robot changes from green, from red to green immediately, there is the possibility that an accident could happen. Or if it is not synchronized, an accident could happen, something like that, okay? It tells us that if this changes, it's going to influence the other, okay? So it states that the results are not due to chance. So it didn't just happen that you found those results or you have those results. They are significant in terms of supporting the theory being investigated. Now, there is some complexity in these hypotheses in that when we talk about significance, we're talking about statistical significance, okay? And I'm sure all of you have heard of the p-value and the probability. So there has to be a certain amount of experiments or samples that are being tested for you to be able to state that the results obtained are significant and were not by chance. If you have only tested one sample, you would not be able to prove the alternate hypothesis because it could be that the result obtained from that one sample was by chance. But if you found the same result after testing 50 different samples, it tells you that it is not by chance, okay? That it is significant and it supports that specific theory, okay? It is the opposite of the alternate hypothesis. So yes, for every study, you will have a null hypothesis and an alternative hypothesis. And at the end of your study, you would be able to accept one hypothesis and reject the other, okay? And it doesn't mean that if you accept the null hypothesis that your study was a waste, okay? It just states that, oh, so we expected this, but study proves that no, it is not according to our expectation. We actually proved the opposite. And I always tell my students that in research, there is never a wrong answer. Not getting a result is still a result, okay? Because we reflect and we analyze the situation and say, why did we not get a result? And there has to be something that has happened because of that specific interaction that we did not get a result, okay? So remember that there's never a no result is, or a negative result is still a result, okay? The alternate hypothesis challenges the status quo. It never contains the equals to sign, okay? It is generally the hypothesis that is believed to be true by the researcher. So in most instances, we would prove the alternate hypothesis to be true, okay? And it normally comes from prior literature studies, but it is only established when the null hypothesis is rejected. Okay, so you can only say that, yes, we accept the alternate hypothesis because of the evidence provided from the experiment. And because of that, we've rejected the null hypothesis. You cannot have that you accepting both the null and the alternate hypothesis. Okay, it doesn't happen. Okay. And often the alternative hypothesis is the desired conclusion of the investigator. Okay. So in the alternate hypothesis, there's also two types of the alternate hypothesis. In that, there can be a directional alternate hypothesis or a non-directional hypothesis. And what do we mean by this? And a directional hypothesis tells us that the null hypothesis is wrong, but it also specifies which parameter would be the one that is either greater than or less than the value specified in the null hypothesis. Okay. If you look at the example below, it says, students who study with classical music will score higher on tests than those who study in silence. So it specifies that they will score a higher grade, okay, than those who study in silence. Whereas a non-directional hypothesis tells us that the null hypothesis is wrong, but it does not predict the parameter of interest is larger or smaller than the reference value. And if you look at the second example, it says that students who study with music will score differently on tests than those who study in silence. It doesn't tell us that they will perform better or will they perform worse. Will they have higher marks or will they have lower marks? It just tells us that they will score differently. Okay, and this could be because there might be such great variations that one individual scored 5 times higher and another individual scored worse off because their attention span differs and that they focused more on music than instead of studying. So they actually needed to be able to study in silence to score a higher grade. Okay, so that is the difference between the two types of alternative hypotheses. The one is very specific. It gives you a value that you can use as a base, okay, or that you can compare from the baseline score, whereas the non-directional does not give you a value that you can use to compare from the baseline. Okay. So what I want you to do is to formulate a hypothesis. You can choose one of the scenarios, you can do all three. I'm going to give you, just see, I'm going to give you 5 minutes to formulate a hypothesis. I know you're very shy and you don't want to speak to me today, but you can type it in the chat and then we will discuss the possible hypothesis for these scenarios. So the first one is, an environmental scientist examines how microplastics impact marine organisms. What would the hypothesis be for this environmental scientist? The second one, a soil scientist investigates the role of microbial diversity in crop productivity. What would your hypothesis be for the soil scientist? And the third one is, a microbiologist studies how bacterial mutations contribute to antibiotic resistance. It's fine if you feel that you cannot formulate a hypothesis at present and you want to work through and do more reading, but I would be happy if you at least attempt to formulate a hypothesis. So it is 10:54 currently. I would say we start the discussion at 11. Would that be sufficient time?
Yes, Doc, I think so. And let me just remind the participants that I have your mics are all allowed. If you have maybe finished and you would want to share with us what you have, you can simply raise your hand, then unmute and talk. Alternatively, you can also text on the chat platform. Thank you. Let's reconvene at 5. Sorry, at 11 o'clock. That is in 5 minutes time. Then we can share what we are having. Thank you.
Right. Okay, we are back. Is there anyone that wants to volunteer the attempt?
All the participants, your mics are allowed. You can raise your hand, unmute, and make your contribution. Or alternatively, just text in the chat box, and then I can read it out in the hearing of the facilitator. Thank you. Doc, maybe on the first one, would it be, I will specify the null hypothesis, then maybe the opposite would be true for the alternative hypothesis. So if I say there's a relationship between, okay, there's no relationship between microplastics and marine organisms, would that suffice?
Yes.
Okay.
So looking at the answer that I've put up there, and you'll see that I don't say that this is the hypothesis, because for each individual, depending on what they are interested in looking at, it's a possible hypothesis. Okay, that this hypothesis is that if the marine organisms ingest microplastics, then they will have bioaccumulation in higher trophic levels, will increase, and it can lead to possible disruptions in metabolic and reproductive functions. So it's very specific in that you would be looking at the changes in the metabolic and reproductive functions. Okay, and you would say that, okay, so if they ingest it, we expect to see changes in their functions. Okay. Then the second one would be that if soil microbial diversity is high, then the agricultural yield will increase because there's improved nutrients cycling in the soil, and there can be enhanced plant resistance to pathogens. So we'll have a better crop because there's improved nutrients in the soil. And this is because there's a higher microbial diversity. There's a lot more microorganisms that are beneficial to the soil in that they provide the nutrients that the plants need to be able to grow better. Okay.
So Doc, here on this one, let's say, for example, just for interest sake on scenario 2, so that could be specified as a alternative hypothesis. Then the opposite, like maybe for example, saying if soil microbiodiversity is low, then agricultural yield will decrease due to or maybe deficiency of nutrient cycling and that kind of to make it the null hypothesis.
Yes.
All right.
And looking at this one, there's another hypothesis that you could come up with, and you can say that it could be that increased microbial diversity can actually have an adverse effect on crop productivity, in that it might be that the microorganisms that are present in the soil utilize the nutrients, so they compete with the plants for the nutrients, and this could lead to reduced crop productivity. So you can see that there's various different scenarios that you can or hypotheses that you can come up with depending on what you are trying to prove. And then the last one would be that if bacteria are exposed to sublethal concentrations of antibiotics, then they will develop resistance more rapidly due to the selective pressure favoring resistant genetic mutations. Okay, so if we administer antibiotics that do not actually kill off the microorganisms, but instead they just give the bacteria an exposure to these antibiotics, these bacteria can then develop mutations that are adaptive to these antibiotics, and they will actually develop resistance.
It would have been very interesting to also hear what the participants have to say. If there's anyone who would like to share with us, you can unmute your mic and share with us, or just type in the chat box as the presentation proceeds. Thank you.
Yes, please do, because you can see that there is no right or wrong hypotheses. We all look at it differently, and I would love to see the angle from which you have looked at the scenario and what you would be interested in formulating as a hypothesis. Is there anyone? Nobody's in the mood today.
Yeah, I saw a hand up. Okay, I think that was Miss Pila talking. Miss Pila, if you can hear me, you can go ahead.
Can you hear me?
Yes, I can hear you.
Okay. Professor, it's Anita, sorry. I just wanted to confirm with you. So on the hypothesis, you have to give reasons why certain things happen that way. For example, you cannot, let's take scenario number one. You cannot just say if there is high microbial diversity, co-production increases. You have to like always give a reason why this happens, and you cannot just make that short statement to say if there is a high microbial diversity, then crop production increases and you say.
Doc,
Sorry, Pila, that
did you
wasn't
hear?
very clear for me.
Yes, yes. Miss Pila, we cannot hear you clearly. I don't know if maybe alternatively you can type, or you can recite the question again for Dr. Miet. Hello, Miss Pila, are you there? It seems
Can you hear me
yes.
now?
Yes,
Yes,
now
we
we can
can.
hear you.
Okay. I was asking Dr. Mita that just to confirm with you, so on the hypothesis, you have to give justification why certain things happen the way that they do. For example, let's take the scenario number one. So you have to say, you cannot just say, let me make an example. If there is high microbial diversity, crop production increases and end it there. You have to always give a reason why things happen the way that they do for you to prove it.
Yes. Yes, that's correct. So remember, your hypothesis has to answer and have the characteristics that we discussed, that it's observable and testable, because how are you going to test that the microbial diversity increases crop productivity? There's something that you have to use to be able to formulate those experiments. So it needs to be observable and testable. It needs to be specific. You should be able to predict the type of tool that you will use to prove your hypothesis. Would that answer your question, Pila?
Okay, very clear. Yes, it does. Thank you.
Anyone from the participants who would like to share something with us? I don't know why the audience was too quiet today, but we can proceed, Doc.
Okay, so we continue. So we now move on to the next step of our discussion, and that is designing an experiment. So now that you've made your observation, you thought of your questions, you formulated your hypothesis, we now need to conduct the actual tests to be able to prove whether our hypothesis is correct or incorrect. Okay, and so an experiment, according to Cambridge, is that it is a test that is done in order to learn something or to discover if something works or is true. Okay, and if you take it on the very basic level, when you cook a meal, you're also conducting an experiment because you're not sure if that meal is going to taste nice or not. Okay, so you are actually experimenting. Okay, so what are the crucial elements when designing an experiment? And I'm not going to give you the information just yet. I want you to give it some thought because I know every one of us has done an experiment at some point in our life, whether it was at school or at in your practicals, in your undergrad studies. We have done an experiment, and I just want you to think of what are the components of an experiment. What do you do when you are conducting an experiment or you're planning to conduct an experiment? There's certain factors that an experiment has to have. We spoke about it in the hypothesis as well. So if I give you an experiment and I tell you that you want to determine the effects of chlorination on bacteria in a water treatment tank, where will you start? How are you going to determine the effect of the chlorination? What are you going to test? Firstly, you would need to formulate a hypothesis, correct?
I was also thinking of saying the same along those lines, that you have to be specific as to which variables are you also going to test there. Which one is the dependent, which one is the explanatory.
Correct. So you gave the keyword, and that is the variable. Okay. So what is the crucial elements that we need? We need to know what the variables are. Okay. So you need to consider that what is the independent variable and what is the dependent variable. Okay. And the independent variable is the variable that can be manipulated. And when we say it can be manipulated, you can either increase the amount, you can decrease the amount, you can remove it completely from the study. That is what we talk about when we say the manipulation of a variable. Okay. And then we have the dependent variable. And this variable is the variable that's going to give us the response. So if you apply 80 tons of pressure on a piece of rock, the independent variable is the pressure, because we can play around with that pressure. We can change it. We can increase the pressure. We can decrease the pressure. We can apply no pressure. And the rock is what is the dependent variable. That's what's going to give us the response. Because if we apply a high amount of pressure, it's going to crush the rock. It's going to break it down into smaller pieces. Whereas if we don't apply any pressure, the rock remains intact. Okay, so can you see, with a simple example, you can already identify what an independent variable is and what a dependent variable is. Okay, and then we have something called control variables, which we're going to speak about. And control variables are those variables that influence the dependent variable response. And these could be environmental factors. So what happens if you applying pressure, but you also adding water to the rock? What is the water going to do? Is it going to speed up the process of that rock breaking up, or is it not going to have an effect? Those are control variables. Okay. What happens if you conduct this experiment and apply pressure on the rock in different temperature settings? Would you get the same result for the experiment conducted in a desert or a high temperature setting versus the Antarctica? Is there going to be an influence? So those are the control variables, okay? And then you will see on the slide, there's something called a control group and an experimental group. And this is the most important part of making your study sound and valid. And that is because the experimental group is easy, right? We all know that that's the group that you are testing. But how do you know that the test that you are doing on that experimental group is correct? That the independent variable is the one that is producing the causal relationship to the dependent variable and not something else. It might be that we have incorrectly identified the independent variable. And to be able to check that, we have a group that is called the control group. And this control group is the group that basically is the baseline that says, okay, so we keep all of our variables the same. And what is the outcome that we see in the control group? Okay, so in the control group, there's an unchanged factor. But in the experimental group, you are changing the factors. Okay, so if we come back to our example on you want to determine the effects of chlorination on bacteria. The independent variable would be the chlorine, okay? Because we want to look at what the effect of the chlorine is. What is the chlorination killing the bacteria, not doing anything to them, making them more resistant, or are they actually thriving and multiplying at a faster pace in the presence of chlorine, or are they dying out? Okay, so that's the independent variable. And the influence that it has on the dependent variable is the survival of the bacteria. Can these bacteria survive? And then if you look at the three arrows in red, those are the environmental factors, okay? Or they could be those external variables that could influence the outcome. The time of exposure, it will influence our result, because if you take the chlorine and you add it to the water and you leave it there only for 2 seconds, you might say that, oh, but we still have 90 bacterial cells growing. But if you take the chlorine and you leave it there for 2 hours and you check, you will say that there's only 10 bacteria that are growing now. So time plays a critical role in influencing our dependent variable as well. Okay, so to eliminate these external influences, we need to make sure that we keep it constant for all of our experiments. So if you want to look at what happens to the bacteria at 2 hours exposure to chlorine, you would take your different types of bacteria, you would expose them to chlorine for the same period of time, and then you will see how many of them survive or how many of them don't. Okay, so these variables, the time, exposure, the concentration of the bacteria and the volume of the water or chlorine can be changed. They can also be manipulated to influence the dependent variable. Okay. So, like we said, the experimental group receives the variable being tested. Okay. And there's manipulation of the variable. We also have something called random assignment. Okay. And this is where you don't say, oh, these bacteria are my favorite bacteria, so I'm going to add them to the experimental group because I know they perform well. They're sensitive to chlorine, so they're all going to die, and I'm going to get good results. No, we need to eliminate the biases, and we need to eliminate the researcher's influence on the experiment, okay? And how we achieve this is by random assignment, where you will say, okay, so I don't know which bacteria are which that are inoculated into the water samples. And you randomly assign, and you say, okay, bottles 1, 2, and 3 will be part of the control group. Bottles 4, 5, and 6 will be part of the experimental group. Or you say, bottle 1, 3, and 5 will be part of the control group, and 2, 4, and 6 will be the experimental group. Sorry.
Dr. Mieta, are we still together?
Yes, I am.
All
I
right,
just
thank
needed
you.
a breather.
Okay,
I've been talking
thank
for
you.
long.
All right, thank you so much.
Okay. Sorry for that. So that is what we mean by random assignment, okay? And the control group does not receive the variable. So the control group, we will not add the chlorine to the control group, okay? Because if we add in chlorine to the control group, then we don't know what the effect of that chlorine is. If we don't add it, we know that the bacteria are supposed to grow. In the absence of chlorine, bacteria are going to continue growing, okay? But there's also different control groups in that we can have a control group where we don't give the treatment, okay? So that is, we're not adding any chlorine. And we read the result, okay? We could also have a placebo control group where we say, okay, so we're not going to add chlorine, but we're going to add another disinfectant to this group, and we see what effect it has on the bacteria. We then have a positive control group where we say we know that we're going to add 5 moles of chlorine and it's going to kill all the bacteria in this sample, okay? So we expect to know that we're not going to find any bacteria growing. Then we have the negative control where we don't add the bacteria, but we can add all the other components. So we can add the chlorine. And we see that what's going to happen? Nothing, okay? There's going to be a negative result. There's going to be no bacteria growing because they were not there in the first place, okay? And then we have something called historical control group. And the historical control group is the one that uses historic data for comparison. And this is used mostly in your qualitative studies, okay, in more the humanities and so forth, where they will look at previous data that was collected, say from interviews or from case reports and so forth, and they will use that as a control for your experimental group, okay? So why do we need the control group? And if you look at the image, you can see that we need it to be able to compare the results that we get from the experimental group, okay? And the difference between the two gives us the evidence that is used to defend our claims and to defend our hypothesis. Okay. Then we come to sample size. So I've touched on it, where we said that the sample size is very important, okay? Because it specifically states the number of experiments that will be conducted or the number of participants in a study. We need to be able to have a suitable sample size. And why is it? A larger sample size will provide more precise results, okay? And it also influences the statistical power, because remember, I touched on this and I said, we normally look at the probability and the significance level of normally a 95% confident interval, okay? We are 95% certain that the results that we have found did not happen by chance. They have some sort of significance, okay? And this statistical power leads to more reliable conclusions. Because if you think about it, if I come and tell you that I tasted 2 biscuits out of a tub, and I tell you all the biscuits in that tub taste wonderful, are you going to believe, or are you going to take my result as concrete and say, yes, it's very reliable, all those biscuits in that container taste brilliant?
That will be doubt, something that one can doubt.
Yes, we cannot, that is not reliable, because you only tasted, say, you only tasted 2 out of a tub of 50 biscuits, okay? But if I come and I tell you that there's 6 types of biscuits in this tub, and I tasted 1 biscuit from each type, and all 6 of those biscuits were very tasty, would you be more convinced?
Yes, yeah.
Okay. So essentially, if you look at it, we do this on a daily basis, because the sample size basically convinces us to be able to accept the findings. And this is very interesting in the sense where we see those adverts of detergents, where they say kills 99.9% of bacteria. And my question is always, how did they test that? Because the chances are they didn't actually test it, because that 1% is always disputable. Okay. An example that I have here, okay, so I gave my biscuit example, but this one, we want to investigate the effect of pH on bacterial diversity in soil. We will collect soil from 5 different sites with different pH levels, okay? And then we will extract bacteria from 5 samples per site. So in total, we will have 25 samples instead of 5 samples, okay? And then we can say, okay, we're taking it a step further to increase our sample size to make it more reliable, is that we're not only going to look at a specific season, we're going to sample in all 4 seasons. And that means we will collect a sample every 3 months. So that would be 25 times 4 samples, which will give us 100 samples. So can you see how we have increased our sample size to make our results more accurate and more precise. Okay. Then another factor that we really need to look at when we're designing our experiments is the terms repeatability and reproducibility. And what do we mean by this? It is very important that our experiments can be done. Firstly, they can be repeated and we will get the same results or very closely related results. And another one would be where I can conduct an experiment, the experiment, and get a set of results. And you can conduct that experiment in your lab and get the same result. Okay, and that is reproducibility. Okay, so we can reproduce those results. Because remember, when we are writing, when we're publishing our findings, we need to give the methodology. Okay, and the reason for that is to show that our study was robust and our experiments were conducted correctly, because another individual in another country, in another laboratory, not a different environment, can take that same methodology and follow it in the same manner that you had done and get the same results. Okay, that is reproducibility. So repeatability measures the variations in measurements taken by a single instrument or person under the same conditions. So what we mean by this is that it should be in the same place, the same measurement procedure, the same observer, the same instrument using the same conditions, and it is repeated over a short period of time. So you keep everything the same. If you are running the experiment, you will run this experiment today, you'll run it tomorrow, you'll run it next week, you'll run it the following week. And the results that you get using that same piece of equipment should be the same or within the same range. Okay, that's what we mean by repeatability. Your experiments are not repeatable if you do the same experiment with the same equipment, but you get two varying results or two polar results, two opposite ends. That tells you that there's something wrong. Okay, if you keep all of these factors the same and you get two totally different results, that tells you that something is incorrect and you need to go and troubleshoot and figure out what could be the reason for this. Okay, because then your results are not repeatable. Okay, reproducible, it measures whether the entire study can be reproduced in its entirety. Okay, so there must be a certain degree of agreement between the results of the experiments conducted by different individuals at different locations with different instruments. So say, for example, I conduct an experiment in the lab and I identify the organism as being an E. coli. And you conduct the experiment in your lab and you also get an E. coli. However, another individual does the same experiment in a different location with different instruments, and he now all of a sudden gets a gram-positive organism, like a staphylococcus. Okay, there is no reproducibility. If you have the same method, but with different individuals, different locations, and different instruments, you should get a certain level of agreement. Okay, and this, I like this picture because it explains it so nicely, in that something can be repeatable and accurate, but it can also be repeatable and not accurate. In that, if you look at the second image, let me find my pointer here. if you look at this first one, it says it is accurate and repeatable. Okay, so we want all of our results to be on the target. And all 4 experiments that you conducted, you got results in the center, okay, on the target, on point. Then it could be repeatable, but not accurate, in that all the results are in the same range, but they're not at the target, which is where they should be. So they're repeatable in that every week that you conducted the experiments, you got the same results in the same range, but they were not what they were supposed to be at the target. Okay. Then we can have those that are accurate, but not repeatable. So they're within the same range here, but there's differences in them, that they're not closely aligned. Okay. Or we have the last one, which is not accurate and it's not repeatable either. They're lying at opposite ends of the spectrum. Okay. Then we have accurate versus reproducible. In that this one, in the first picture, it's not accurate and it's not reproducible, because you can see there's completely varying results. Okay. Then this one is that it's reproducible, but it's not accurate. In that if you use different pieces of equipment, you got the similar result, but it was the similar incorrect result. Okay, all the times that you did it. And then the last one is where it is reproducible and accurate. So that is what it should be. And each time that an individual did it, a different individual did it with different equipment, they all got a result in this range. Okay. And this is what we aim. So we want our results to be reproducible, like our experiments, they must be reproducible and they must be accurate and repeatable as well. If you achieve this, then your experiments have been credible and they are worthy to prove your hypothesis true or false. Okay. Now we have different types of experimental designs. Okay. So it's not just about, oh, okay, so I'm going to do an experiment and conduct it 3 times over 3 weeks, and I'm going to get my friend to do it at his lab and done. And I'm going to use those results. No, there are different types of experimental design. And we have something called the pre-experimental research design, where a group or various groups are kept under observation after some factors are recognized for the cause and effect. Okay. And then we have a true experimental research design, which is what most of our experiments are. Okay. It is the only method that establishes the cause and effect relationship within the group. Okay. And then we have a quasi-experimental research design, where the participants of the groups are not randomly assigned. So the researcher cannot make a cause or effect conclusion. And we're going to go into more detail on this. What we mean by a pre-experimental design is that there are 2 different ways in doing a pre-experimental design. And you would normally do a pre-experimental design in preliminary studies, okay, where you have little control over the variables and you want to see that, is it actually viable to do this experiment? You will do a single group, okay? So you can do a one group pre-test, post-test design, where you take a single group and you test it before and after a treatment, and that's it. You don't have a control group to be able to compare, okay? Then you have a one-shot case study, where you say a single group receives a treatment and it is then is tested, okay? So there's no pre-test or no control group. And this can be, say, for example, you have a new training program for your staff and you make all of your staff do this program, and then you measure their productivity after the program. But you didn't measure what their productivity was before they undertook this training program. Okay, it might be that they were fairly productive, and then you gave them the training, and it pushed up their productivity by 10%. Or did this training program have no effect? If they were unproductive, they're still unproductive after the training, because it's their attitude that actually plays a role in their productivity. Okay, so the intervention that you put in or the treatment that you provided was not correct and was not effective. Okay, so we normally do this for preliminary studies. Okay. Then we come to the true experimental design. And this is the pre-test, post-test control group design. Okay? And this is where most of our natural sciences experiments come in, and they use the pre-test, post-test control group design. Okay, where participants are randomly assigned to either an experimental or control group. Both the groups take a pre-test before the treatment. Okay, so for example, say you want to test the effect of a new drug on blood pressure. Okay, you have a group of 40 individuals in a room. You randomly divide them into two groups. So you randomly divide them into the two groups, and you take both the groups' blood pressure, right? You note it down. Then you give the one group the medication, and the other group, you don't give them the medication. Okay? And at the end of that treatment, you then retake the blood pressure for both the groups. So you will check if the blood pressure was the same for the control group, because they didn't have any treatment. Okay? So their blood pressure should remain the same. Versus the experimental group, if they had extremely high blood pressure and you gave them the medication, and then you saw that in the post-test, when you took their blood pressure again, their blood pressure decreased. Okay, so you can then use these results to make your conclusions, to do your analysis and derive that yes, this drug is effective in reducing the blood pressure of individuals, or no, it does not reduce the blood pressure of individuals. Okay. The second type of the true experimental design is the post-test only control group design, where participants are randomly assigned to groups. You don't take readings before the treatment. So you divide them into two groups, you give the treatment or you put in the intervention, and this you only give it to the experimental group while the control group doesn't have it. And then you test the outcome on both the groups. So that's the post-test. Okay, so there's no pre-test, it's only a post-test. So for example, you want to measure the performance on a test after some students get tutoring assistance and others don't get tutoring assistance. Okay, but you did not check what the performance was of this, both the experimental group and the control group, before the experimental group got the tutoring. Okay, so you don't know what those marks were. Okay. And then the third type of experimental design that you could use for the true experimental design is factorial design. Okay, and the factorial design is where you test two or more independent variables at the same time. And groups receive different combinations of factors. Okay, so this is a more complex experimental design in that you don't only have one independent variable that you are testing, you are testing numerous independent variables at the same time. So for each variable, you would have a control and an experimental group. Okay, and you want to then see the difference or the effect of each of those variables on that experimental and control group. Okay, so say, for example, you want to study how diet, a low protein diet and a high protein diet, and exercise, so individuals that exercise daily and those that don't exercise at all, what's that effect on weight loss? So you're going to have different groups, correct? Where you're going to administer a low protein diet with no exercise to one group. You will have a second group where you have a high protein diet with no exercise to the second group. The third group will have a low protein diet with daily exercise, and your fourth group will have a high protein diet with daily exercise. And you measure what all of those combinations will have, or what effect they will have on the weight loss. Okay. Then the third type is the quasi-experimental. And this is the non-equivalent control group design, where like the pre-test, post-test design, we have the same design, but these groups are not randomly assigned. Okay, so these groups, you specifically put in certain individuals in a group because you want to see a specific outcome. Okay. Then the second one for quasi-experimental is time series design, where you have repeated observation before and after treatment. And this helps track changes over time, especially when you want to look at, say, your factorial setup, where you looking at different variables, independent variables, over a period of time. So an individual who has a low protein diet with daily exercise over a 6-month period, is their weight loss greater than an individual who has a high protein diet with daily exercise over a 6-month period? Okay, so these are time series designs. So basically, all of that in a nutshell, put together. You develop your experimental question or hypothesis, okay? You define your variables. What are the treatments? What are the controls? And what are the responses, okay? You then define your experimental and sample units, okay? You estimate your sample size. So it's not a random estimation. Most of our sample sizes,
Dr.
Okay.
Thank you.
Sorry for that. So most of our sample sizes are estimated using a calculation, okay? And this calculation ties in with the statistical significance. So like I always tell my students, that if you are using or you're conducting a study or you want to analyze results from data that has already been collected, you would need a minimum of 20 or 50 samples, 50 data points, to be able to make a substantial conclusion. okay, to be able to do statistical tests to tell your story, okay, and to support your hypothesis, okay. And then randomization and layout, which is how you are going to do this. How are you going to conduct your experiments? Are you going to do them for only a month every day, or are you going to do it over a 6-month period, once a week? So that would be what your layout and randomization is. And this here basically speaks to our repeatability and reproducibility. Okay. So now that we've said a whole lot and a mouthful, I don't know if you want to do this now, or do you want to take down this hypothesis and then attempt it on your own?
Can we hear from the audience or from the participants? And the participants have been very quiet throughout the session.
It's either they know all of this and I've bored them, or they are totally lost.
Yeah, but I think this was a very, very insightful conversation that we had. It was a thorough presentation. I will also talk with the team. I think the title, we should change it. Because for me, this was not just for natural sciences, but it was for both social and natural sciences. I know that we define our approach, in terms of that in social sciences, we cannot, subject human beings to experiments. You cannot subject them to poverty. You have a control group, then another group is the experimental group, but we share a lot in common. And I was so, I noticed some striking similarities in the way in which we formulate hypotheses, as well as designing our experiments as well, because we have social experiments, so that we also perform. So this was supposed to be for both natural and social sciences. Thank you so much for a very insightful presentation. I think I saw some of the things that I also talk to students when they come and do consultations here at the RCD in terms of results. Even if you get negative results, they are still results. Even positive results are still results. If you reject the null hypothesis, it's still a result. If you accept it, it's still a result. But students, in some cases, they tend to what they want results that confirm their a priori expectations or what they were thinking or the hypothesis that they have posed. So in the end, they end up, there's this saying that if you torture the data, it will confess. So they end up manipulating or massaging the data so that it can speak to their conscience or what they want the data to reveal. So thank you so much for the presentation and for agreeing to facilitate this session. I think we can open the floor for questions, contributions, and general discussions. The audience, I think all your mics, let me cross check again, but I think all your mics have been allowed, and you can just raise your hand, unmute yourself, and then share with us or ask your question if you have any question. So all your mics are allowed.
Yes, I know the session is being recorded. So if anyone wants to go and design an experiment using the following 2 hypotheses, you will see that there is a discussion on your experimental design.
It seems, Dr. Mehta, that there's no questions from the audience. There's no comments as well from the audience. I would like to take this opportunity to once again express our appreciation to the participants and also to you, our facilitator for today, for agreeing and taking your time to prepare and facilitate today's workshop. So I think we have been equipped with quite a number of tools that will sort of enhance our approach in formulating hypotheses and also in coming up with research, experimental research designs. Or designing an experiment if you are from the natural sciences. I also want to remind the participants to complete the feedback form that I've posted there in the chat for your access. By filling the survey form, this also ensures that you receive a certificate of attendance. And as I have alluded to earlier, the recording and the presentation from this session will be uploaded on our Moodle community under the workshop information and resources tab in about 3 to 5 working days after we've reviewed the content for suitability. Then also, if you need more information about us, you can visit us on www.uj.ac.za/postgraduate school/research capacity development. Or for any queries, you can contact the RCG inquiries at uj.ac.za. I also want to bring it to your attention that we recently launched the Research Capacity Development Management System. So if you missed that launch, this is a system that we are now using as the RCD to streamline our events and offerings, meaning the services that we offer. And on this platform, you can register for workshops, research writing consultations, and stay updated on all the events and services that we offer as the RCD, all in one place. Hence, we label it as a one-stop access for all your events and services. So we plead with you to register and utilize this system. It's accessed through the U-Link under the services tab, or you can visit the rcdms.uj.ac.za for you to be able to register. So let me once again check on the chat to see if there's any comments or any questions. I see okay, DG is typing a question. I don't know whether it's a question or it's a contribution. Let's just await that. Okay. You are indicating that the assessment form is yet to be posted here. So I have posted the feedback form. It's just right above. I think just the message, the second message after the one that you have posted to Baba today. Okay, DG. I think I've pronounced your name correctly. All right. So I think if we do not have any questions from the participants, allow me, Dr. Mieta, to end this session. If there's any words that you may like to leave the participants with, just as your final remarks.
No, thank you so much for giving me the opportunity to present at this workshop. I would really have loved a lot more interaction, but we hope that the students have taken the information provided and are able to implement it and utilize it in their studies. And yeah, should you have any questions, I know I have interacted with the postgraduate school previously, and it is a brilliant community to be able to work through your postgraduate studies, especially individuals doing doctoral studies. It does get very lonely, so make use of the community that UJ is providing you the opportunity to utilize. Thank you very much once again, and I hope everyone has a lovely day further and a break if you are taking a recess period, or being a postgraduate, we generally work through recess as well.
Thank you so much, Dr. Mehta. And once again, I thank you for facilitating this session. And to the participants, if you are not yet enrolled to our Moodle community, you can email us at rcdinquiries at uj.ac.za. Let me just put it in the chat. RCD inquiries at uj.ac.za. So I've posted the email that we use for inquiries, so you can just give us your information, the level of qualification that you are doing, and also just give us your student number. Yeah, I think with that information, we'll be able to enroll you on our Moodle community so that you can have access to past resources, for workshops and events that we have held, and also for you to receive communication on our workshop and other offerings. So with this, allow me to end this session and wishing you a great day further. Thank you for continuing to support the RCD. Thank you once again.
Thank you.
All right. I'm now ending this session and concluding the recording for the session. Thank you.